Hello, everybody, and welcome to today's webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I'm hosting today's session on AMR R&D efforts in the CMC and formulation arena. Do it right the first time. We're pleased to have developed this webinar in collaboration with the Repair Impact Fund. Revive is GARDP's education and outreach program and was officially launched in 2018. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All webinars are recorded and can be viewed after the live broadcast on our website, revive.gardp.org/webinars. As well as our webinar recordings, you can also read our series of articles known as Antimicrobial Viewpoints, where experts in the field discuss various topics within. At least our latest contribution was one from Matthew Leveson on Pyramid AMR. We also have a resources section that includes the Antimicrobial Encyclopedia. This has definitions on various important terms, and some of these include videos where experts give a further explanation. This resource will soon be expanded with new terms and can also provide suggestions to us at revive@gardp.org. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar via the questions window in your webinar control panel, as shown on this slide. We will address the questions after the presentation, and we'll do our best to respond to as many as possible. Our moderator today is Michael Scholl. Michael is the Chief Executive Officer of Leukocare AG. Welcome, Michael. I now hand over to you to introduce today's speaker.
Thank you, Victor. I think everybody should be able to see and hear me now. It's my pleasure and honor to moderate this exciting webinar today. And I think we all can look forward to the talk of our today's expert, Aleksandar Danilovski, about some strategies to fight AMR. A quick introduction to Aleksandar. Aleksandar Danilovski, I'm proud to call him Sasha, is Chief Scientific Officer at Xellia Pharmaceuticals in Denmark. Moreover, he's also a member of the Scientific Selection Board of Novo Holdings Repair Impact Fund and a board member with companies in Sweden and Denmark, namely Active Biotech in Sweden and Formulation in Denmark. Previously, he has been head of European R&D for Bayer Laboratories and also member of the management board with Cleaver and a member of the board at Rutgers Innovation, Boscovich Innovation. Sasha has received his PhD in chemistry from the universities of Cambridge in UK, as well as the University of Zagreb in Croatia. And please allow me a few personal words. I know Sasha for a while in his capacities, and he's not only an outstanding scientist, but even more importantly, an inspiring manager to his team, and also a great personality. And his motivation to provide better antimicrobial therapies to patients and physicians is purely intrinsic. So we all can be very happy to have dedicated experts in this important field, as Sasha is one. So I'm honored to call him a dear friend. Today, Sasha will talk to us about the challenges of antimicrobial resistance and strategies that can help us to tackle these challenges. Beyond perspectives on new therapeutic approaches, he will emphasize on the potential impact of CMC strategies and CMC, and in particular formulations providing improved characteristics to antimicrobial products can enable value-added products for better treatments. So I think one aspect is to avoid pitfalls during clinical program. The mantra should be do it right first time. And having said this, we all can look forward to hear what Sasha's telling us about this field in this respect. And with this, Sasha, I hand over to you and the stage is yours.
Thank you, Michael. I hope everybody can see and hear me. I would welcome you all and would say, not knowing who we have as a participant, so good morning, good afternoon, and good evening to you all. And thanks for joining into this webinar. I would like to start to thank Victor, the GARDP team, as well as my colleagues in Repair for putting together this very interesting series of online webinars, which are trying to shed a bit more light on a very important healthcare topic, which is AMR, or antimicrobial resistance. So idea of today is that I would like to, it would be much easier, by the way, if we would be in person, because for the ones that know me, it would be quite interactive and I would seek for your engagement during the presentation. This time around, we will do it in an online way, so we will answer the questions at the end. But please bear with me. I will try to be provocative at some points, with deliberately provocative, and seek also your critical thinking and reflections on something which is an important topic. So antimicrobial resistance, if we move on. Okay, so we will start by setting the scene, and it will revolve mainly around AMR. And then I would like to go into some of the considerations which are not typically your first choice when you are talking about AMR R&D. It's usually the compounds, its biological activity, and all the also properties related to in vitro and vivo profiling, which is extremely important. And it all starts with that. But I would just start to broaden it a bit and add flavors associated with CMC and formulation, and also link it a bit with its relevance in the bigger scheme of things. So let's start. This has nothing to do with AMR, but it has to do with basically what I'm asking you for. And I'm asking you for all of us as scientists, as people that are working in the field, and not necessarily scientists, also policymakers, also healthcare professionals, that we really need to change the way we think. And as everything in life, many things have been said way back in time. So also this famous quote for Albert Einstein is saying it much better than I can do. AMR is a problem, it's a healthcare problem, and we will not be able to change it as a holistic problem if we don't change the way how we think. And the relevance of that is also linked to the human psychology or behavioral psychology. And Amos Tversky and Hal Sachs are quite well-known behavioral psychologists, which were doing a lot of interesting studies about, how not just the way of thinking, but how you are approaching things is influencing the outcome. So if we take this very simple example, which is when they did research and in the medical field, in the area of surgery, and then when surgeons were talking to the patients, as we all know, you need a consent to a surgical procedure. When the basically the doctor that was doing the surgery, the surgeon, was focusing before the consent on the operation success, 82 of patients said yes to the operation, only 18% declined. When it was turned upside down, which is mathematically still correct, because if 90% is a success, then obviously 10% is not success or failure. But when the surgeon focused on the potential failures and the statistics of the failures, you can see how dramatically the response from a patient changed. So from 82, the yes dropped to 54, and no increased accordingly from 18 to 46. So this very powerful, at least for me, takeaway is that it's same situation. Left and right hand side are completely the same. The mathematics is, it's the same situation we are describing, the same sets of circumstances, but by the way how we are approach it, by the way how we think, we absolutely can impact and change the outcome. And this is what I'm wanting to achieve today, to shed light on some of the things which are linked to the how we think, and also let's try to think differently. Here, this is not yes or no, positive, negative, but I'm just alluding to let's try to change how we think, because by that we will definitely change the outcome. Antimicrobial resistance is a problem, and I'm sure everybody on this call would acknowledge that and is aware of that. And this is this slide is basically taken by one of the very first holistic report done by on the topic of AMR by Jim O'Neill. I believe it was 7 years ago, around 2014. And in that great report, which I urge everyone, if you didn't read it so far, to do that. It's a very nice and very rounded, holistic thinking around the subject. And Jim O'Neill is an economist, so he basically said it's a very simple equation how we can fix the AMR problem. And it was done, by the way, just last comment, yes, Jim O'Neill was the coordinating factor, but it was a very multifaceted approach, and in the team there were people from across the space, from healthcare and other areas. And he basically concluded with the team, it's very simple. We should lower the demand and increase the supply. It's quite an economical way of how you can approach the challenge. So we will fix the AMR problem by lowering the demand. Lowering the demand would mean we should basically lower and have more controlled use of antimicrobials. And I really don't want to go into all the details because this is a topic in itself. But this is important that we know that lowering the demand should be done in the way of how we look at One Health. One Health is a paradigm that brings together three aspects of health: human health, animal health, and on top you have environmental health. So by combining environmental, human, and animal aspects, you have a one coherent story and One Health, and lowering the demand of antibiotics should be in the scope and in the context of One Health. As a good consequence of that, now these days quite more and more and quite more responsibly, antimicrobial or antibiotic stewardship has been put in place, which is the medical practice, and very good medical practice, which says that antibiotics should be prescribed when and if needed, and not in the case where we don't know what the causative pathogen is, like in the viral infections, you don't need to give antibiotics and stuff like that. By doing this responsible use of antibiotics in the One Health context, we will clearly lower the demand. There are also some other elements of, believe it or not, wash our hands, which we also see these days in the current pandemic, which is not an AMR issue, but it's a viral pandemic of COVID-19, how some basic stuff, some basic rules of public health are extremely powerful. And on the right-hand side, we have the increasing the supply, and increasing the supply just says we need more drugs. We need more effective drugs that will work in the anti-infective space. And this is where one of the biggest challenges lies, because since probably 10, 15 years, a lot of companies left the field of antibiotic or antimicrobial space, and we have fewer and fewer compounds, and especially even the ones that get on the market are not successful on the market for myriad of reasons. And that's why we need jointly to work together on push incentives, which are currently quite, I would say, in a much better shape than they were some 5 odd years ago. And push incentives means supporting the R&D initiatives. And now you have various funds in place, from CARBEX, Repair. Recently, AMR Action Fund has been put in place. Also, GARD-P is quite active in this environment. but on the pull side, and pull side means revising and in a way restructuring the economic model and the reimbursement model of the antibiotics is something which we are still lagging behind. And we need to jointly work together as a global society and try to improve that, because we really shouldn't have news like we used to have. And unfortunately, just last week there was another week of another company working in the antibiotic space throwing the towel in and basically calling it a day and completely redirecting the scope. So that's our space. And just to again not go into a lot of details, we really need new drugs, new anti-infective drugs, new antibiotics predominantly, that will work on these resistant strains, because we in a way, if we go just one step back, the first real pharmaceuticals were antibiotics. If we go back to the World War II and introduction of penicillins in the medical practice, especially in the warfare environment, and then from then on the whole pharmaceutical industry just basically built upon that. Antibiotics are extremely valuable drugs, are very much needed, and they are vital for humanity. And unfortunately, right now we don't have enough of them, especially for the resistant bacteria. The reasons are that there is, as I briefly touched, no viable market. There we have investments in the space, especially at a large stage, which are limited. AMR Action Fund will help here, but we still need more. And we definitely need a change in a business model. As one of them, I would like here really to quote Dame Sally Davis, who is very, very prominent advocate of the importance of this topic. She basically summarized it brilliantly. And the quote says that COVID-19, that we are all living for now over a year globally, is like dropping a lobster into a boiling water. You instantly know that you have a problem. In our case, it's the opposite. We are gradually heating the water where lobster is in, and by the time lobster figures out that there is a problem, basically there will be no more lobster alive. So it's a slow and slow-moving problem, but it's of a great impact, and we definitely need to fix it, because we don't want to be in a situation that when we are afraid going to a simple to hospital for a simple surgical procedure, because if we pick up some infection in the hospital environment, that then we will be in a big problem. So with that in mind, that sets the whole scene of the AMR Innovation Challenge. I really don't want to go into the big details of this slide. It's quite a busy one, but we all know it all starts, if we now focus on the R&D efforts and new antibiotics that we desperately need, it all starts and ends with new compounds. And we definitely need more and more fruitful antibiotic pipeline, not just early stage, I would say especially late stage antibiotic pipeline that would have different modalities, different different modalities, different classes of compounds, because we need a very wide armament of options to be able to effectively fight the AMR medical challenge. But besides compounds, which of course need to be active and need to be biologically profiled in vitro, in vivo, and also at later stages in clinical development, and they need to be done so successfully, we cannot forget the one element, and that's the CMC. CMC is chemistry, manufacturing, and control as an abbreviation. It's basically how you make the drug substance and especially the drug product. What I will try to show to you later on in this presentation is that many people overlook this and underestimate the relevance of this quite basic stuff, because it has nothing to do with profiling of compounds, finding new active compounds. It's quite down to earth. But if we don't appreciate that as a potential challenge, if we don't address those elements, we can be very sorry at the end of the day. So we shouldn't treat that as I called it, a foster child. and that it doesn't become theoretical. And I'm a big fan of storytelling and case studies. I would start with showing you case studies and real-life examples that will, in a way, speak more than speak for itself. So if we start now, this is a real case study from quite a long time ago, and it's a discovery program of a new chemical entity way before, not way before, a little bit before 2000. The proprietor of that compound was a big pharma. It was a compound which was of a broad spectrum in nature, and if we all know, before 2000 that was pretty much how all the compounds were. It was actually a derivative of a known class of compounds, and it was in the lead optimization step. So this specific compound at stake here was meeting all the criteria that were defined in the target product profile. So all the in vitro and in vivo criteria, all the MICs on primary, secondary panel of relevant bacteria. Also, in vivo animal efficacy in 2 relevant models criteria were reached. And compound was, because of that, how it goes in a discovery funnel, it was progressed to the next step, and the next step was more detailed pharmacokinetic and safety profiling, which was needed to define the preclinical candidate, or actually defining several preclinical candidates and then moving on further. And as you know, you need more compound for that. So all that was made before was consumed. So for the further profiling, more compounds was needed. And then when the chemists made the new material based on the route of synthesis and on all the procedures that were well described, the compound simply failed on in vitro assays. It started with failing in vitro assays, but not small failures. We are talking sizable failures. So like the original compound had like MICs on the relevant panel way below 0.5, even much lower. The newly made compound had values on those strains all above 16. And also, compound was not made just once. It was done in triplicates by 3 different, even not 3, 2 different groups in triplicates, and the results were confirmed. Unfortunately, Murphy's way confirmed, so not the good numbers, but the bad numbers. So 3 times made compound had MIC values above 16. And this was a big surprise, and of course, obviously not the good news. And then the company decided to, let's say, do the arbitration, if you will, quote unquote, and the final proof should come from the third party, and third party on both chemistry and biology. So then the contracted lab made a compound, tested the compound, and unfortunately the contracted lab confirmed what was done in triplicates before that internally. Because of that, it was obvious that this compound was nowhere near to progress to the next step. You basically, the company needed to go back to the drawing board, back to the lead identification, even if not before, because this wasn't a lead. This was something that had a good results. And company luckily had enough resources and time to do a detailed and lengthy root cause analysis. And they have identified the culprit. So what was actually active in the original compound was not the compound itself. It was a very, very potent solvent impurity, which was, by the way, toxic. And it killed everything, including bacteria. And this wasn't part of any specification. And once the compound was made again, because it was done in a bigger scale, obviously groups of that do compounds in bigger scale are closer to the commercial environment, not commercial, but still how they approach specifications and selection of starting material is slightly more, let's say, stringent. And the solvent that they have used, of course, didn't have that. Clearly the compound made was inactive. Consequentially, all activities was stopped and project was killed. So this is case study number 1. The case study number 2 is a more recent one. It was from a few years ago. and it was from a different company. It was a small biotech. Small biotech that was formed, which is almost very usual way of forming smaller biotechs, as a spin-off from the university. Here the project was around a narrow spectrum antibiotic, novel compound, novel mode of action, and incomparable stage a little bit earlier, so still in between lead identification and optimization, but not that far from where the previous compound was. Again, this compound here met all the requirements in the TPP, and as in previous case, it was agreed to progress to the next step, and as in previous case, more API was needed. Because this was a small biotech company, they didn't have, like in the previous case, their own group who can do the bigger scale. They contracted a third-party CMO, and that third-party CMO started with unreeling millions of problems. First of all, they couldn't make a compound, period. So when they started with a synthetic procedure that was described in the lab notebooks, they couldn't make the compound. To make the problem even more pronounced, the person that was responsible for making the compound, which was a postdoc student, left the university. And the documentation was basically what was written in a paper, but there was no more direct person to talk to. Luckily, because it's a field of expertise of CMO managed to reinvent the recipe and make the compound by modifying quite a lot the synthetic procedure. But unfortunately, when they ended up with the final product, the product was again inactive. This was confirmed by another CMO, and company was in a stage that basically needed to stop all activities. They killed the project, and basically company went belly up. It's bankrupt and liquidated. And they didn't have time to do the detailed and lengthy root cause analysis in this case because there was no company left. But obviously, what was done in the first step and the synthetic procedures that were described initially, and the compound that was made initially was made by this postdoc student, and all the initial profiling was done by that very compound. Once no material was left, once you needed to make new, it was simply not possible to have an active compound. So what are these two case studies teaching us, and what are the key learnings? GMP is and I heard a great abbreviation from people that really consider GMP as a pain in the neck, that GMP as a good manufacturing practice is not good manufacturing practice, but it's actually a great mountain of paper. But nothing can be more false than that, because GMP is something that is, of course, at appropriate level for appropriate stage of the project, is something which is an enabler, which is a gatekeeper, that what you have at hand will be, when you need it, reproducible, that you will have some quality attributes set, and that will enable you that you can make compound more and more of a compound if and when you need it. And also when you come to the clinical development phase, where you will definitely need more material, and that material needs to be made in the full GMP from the real compliance GMP standpoint, that you will be able to make it. So here, the takeaway in the first example was that the quality of starting material was absolutely uncontrolled. Second case was an example where the synthetic procedures and recipes were, let's be polite, were suboptimal. Let's not use the word what they really were. And also the analytical procedures and overall documentation, irrespective of where you are, should be at a quality that you can, when project goes from A to B and a group B picks up the project from group A, that they are able to move on. So I'm not saying here that the real industrial full commercial production GMP should be applied for the discovery project. Not at all, because that is impossible. But what is possible is the way of thinking. And that's coming back to the first slide. We need to think in a way that what we do will need to be repeated by someone, that someone would need to be able to move the project to the next phase, and then we need to document the relevant information. We need to define as much as possible quality attributes and do it right the first time. And I think these two case studies were a perfect example where project failed in the first case, in the second case, not only that project failed, but the company basically got liquidated. And the reason was the manufacturing procedures and the quality attributes of that. And this is a circle of life, and I really don't want to go into details, but I think these pictures of potato cutting are also a nice example what it means. So if you look at the bottom picture, and I don't know if you can see this potato, it's clearly peeled potato. But if you are making dinner, I'm not sure you are happy if somebody peels the potatoes like that, because out of 1 kilo, you will probably end up with maybe 200-300 grams by the time this person who is peeling the potato is done. It's actually not peeling, it's just chopping it off. So it is relevant, and it's relevant from many, many angles. Now moving to another level, and this is a formulation or a drug product. So very briefly, drug product is, if we go here, is when you move from the drug substance into a drug product or a drug formulation. What do? You add a magic compounds to your drug product, and these magic compounds are called excipients. They are, by definition, should be pharmacologically inactive compounds, which are added to the drug substance, and those excipients have a crucial role in the formulation, and they either are behaving as a functional excipient, which means they are contributing to the drug delivery, and that in turn means that these excipients are controlling the rate, the amount of drug, of the substance that gets released into the human organism. And the second group of excipients, which are also very relevant, are the non-functional excipients. These are added not to control the drug delivery, but to basically help you out during the manufacturing process and to also control the drug stability. I will not go into details of what excipients are, but these are the compounds that are extremely relevant, and they are a core of any formulation. So if you, for example, just to be very graphical, take any tablet and put it on a scale, and let's say you have a 0.5 gram dose in there, probably your tablet will weigh more than 2 grams. Of course, it varies product by product, case by case, but let's be quite often it will be about 2 grams, which means you have more than 1.5 grams of stuff in that tablet, which is not your drug substance. And that stuff in your tablet are excipients. So they are, in many ways, predominant mass in any formulation. And that's why we need to understand how they interact with the drug. And how the formulation development is basically done, especially in the initial phase when the composition of formulation is defined. And as I said on the previous slide, formulations typically don't have one excipient. They have multiple excipients. Sometimes, yes, it can be 1, can be 2, can be 3, can be 5, can be 7 for that matter. Just, for example, if you look at the COVID vaccines, in the case of mRNA vaccines of Pfizer and Moderna, these are nano-liposomal formulations, and they have 5 different lipid molecules which make the liposomal core where mRNA is encapsulated in. And how you find those excipients and how you pick which ones you will use, remembering that some are functional, some are non-functional. So for especially the functional ones, you are using the same thing as you do when you are discovering compounds in your drug discovery, in your hit identification. So formulation screening in many ways could be seen as a hit identification. We know from the chemistry hit identification phase that we basically try to find compounds that will bind to a target, and that will bind to a target selectively, and specifically and with a good affinity. And how we are trying to model this, especially, for example, if we have the crystal structure of the target, we can do a lot of the structure-based drug design approaches where we are modeling the non-covalent interactions between the drug and the target, like in this case. In the formulation screening, you are doing exactly the same. You are modeling the non-covalent interactions between the drug and not the target, but the excipients, either one or multiple. And this is basically how you are doing your systematic and rational formulation screening in quite analogous way to how you do the basic hit identification steps. So you are designing the space of non-covalent interaction between the drug and the excipient. Do not going into details, but this is a nice snapshot which tells you, and I don't know if you can see the colors, but this is when the guys in 2016 looked at all the dosage forms in the world, and then they grouped them in the categories and how many are in which category. So for example, oral is a little bit below 20%. Parenteral are 44%. Then you have inhalation, 9%, and so on. So pretty much almost 2/3 of all formulations that are currently sold on the market are either oral, which mean tablet capsules mainly, or they are injectable. And out of the two, injectable are bigger than oral. This is quite similar if you look at this bottom chart. This was a very nice overview of preclinical antibiotic pipeline published in Nature Reviews in 2019, where they clustered, I think, a bit more than 200 different or 300 different preclinical assets into different categories. In the article, there are many different elements, but when you look at the formulation snapshot, and you can see that parenteral, again, is the biggest chunk. It's 43%, almost exactly the same as here. And then oral should be green. Yeah, so it's a little bit more than 50% is, again, oral and injectable. In antibiotic space, there is a lot of topical, which is all your dermal products for very known reasons. But clearly, oral formulations and mainly injectable formulations are the most abundant drug formulations in the world. And then when you look at here the breakdown of therapeutic area, our space infection is in golden middle. So clearly you have a oncology, 1/4. This is CNS, almost another 1/4. Then the blue is diabetes. And then the middle is spread with all the other therapeutic areas where our antibiotic space sits. When this is looked at on a forecasted level, you can see that the value of the drug formulation space on drug delivery systems was in 16 around $500 million. It's projected to double in by 2026. And again, you can see the distribution between the region, nothing spectacular here, quite usual suspects. US 1/3, 1/3 is Europe, and then a little bit less than 20% comes from Asia Pacific. What is this telling us? That basically these two groups, mainly injectable and the oral, are where the majority of current drug products sits. And this is a very busy slide, which I will just very briefly go through, but where we need to, and what matters when you are defining your drug delivery systems is here, up there. And your main strategic drivers is that with formulation, as I said, because it has the functional excipients, which are impacting how the drug is delivered into the body, by doing the proper formulation, you can do a lot of stuff. So if you have a drug compound, which for example has a quite not favorable safety profile, which means it has a lot of safety concerns, by proper formulation, you can make that drug more affordable. For example, amphotericin is a drug which is quite effective in the antifungal space, but not necessarily with the best safety profile. But when Gilead did the liposomal amphotericin, it completely changed the market, because liposomal amphotericin is extremely safe. And it is safe because of the liposomal formulation. So by formulation, you can minimize the side effects. You can also improve the drug delivery in the target area, which means you can deliver the drug where it needs to be by some functional excipients which will go where you want drug to be released. You can also improve the patient experience, the quality of life of patients. For example, who wants to take drug 6 times a day? Or 3 times a day if you can take it once a day, or even once a week, or there are some formulations which are even once a month. All those effects are achieved by the means of formulation, not by the means of drug substance. And that's why it is extremely relevant area to be aware of. And as said, this just says it again, by formulating a product, you can impact 3. If you cluster this busy slide, you can impact 3 areas. You can improve safety, like the example of amphotericin, and some more examples are coming our way. You can also play with the efficacy. You can modulate PK/PD, for example, lower the dose, change the dosing regime and stuff like that. And you can, which is also very important and we should never forget, we are in this business because of patients. We are here because we need and want to improve patient lives. And improving patient experience, patient compliance, and the quality of patient lives makes it's everything. Because we all have parents, grandparents, and we all know how taking drugs is especially if they take multiple drugs, is not an easy task. And whatever we can do from the science side to formulate products in a more patient-friendly way, we are doing a lot, and we are making everyday life of a patient in need much, much better. So these are the 3 main goals we want to achieve. And if you then ask why, besides the main reason, which should be making patient experience better and improving patient lives, you are also, believe it or not, rewarded on the market. And I will show you very briefly 3 live examples. And it's from the 3 different molecules. Molecule number 1 is irinotecan, a very well-known oncology drug. And this is, and I don't know if you can see anything, I'm on my screen, it's quite small, but nevertheless, it's a product where this snapshot is showing it. You have the brand, which is Compostar, you have 10 generics of that brand, and then you have a liposomal formulation of the irinotecan. And if you look on the volume, so the gray, if the 2017 snapshot, are generics. So generics are having, I would say, more than 80 or 85% of the units that are sold. The blue, the pink, or the orange is the brand, which is 10%, and only less than 10%, or a little bit shy of 5%, is the liposomal formulation when it comes to volume. But look what happens when you look at the dollar value. The blue is 3/4. So the Onivid, or the liposomal irinotecan, is basically 75% of the market. And look at the unit price. It's more than 10 times more expensive than the generic. So the brand is a $35 a vial. Generics are around $13 a vial, and liposomal irinotecan is around a little bit less than $1,500 a vial, and it's leading the market. Example number 2, liposomal doxorubicin. Again, generics, liposomal generics. The price differential here is less. I think it's around 4 times. So the doxorubicin as a liposomal doxorubicin is around 4 times more expensive than the generic generic doxorubicin, the plain doxorubicin vial. And still, even with that price premium, they are having the dominant market position. And a very well-known example is Abraxane, or the nanoparticle albumin-bound paclitaxel, where you can see here, here the volume and values are, by my mistake, swapped, but the volume is here. So Abraxane is around 40-45%. That's the improved formulation. The 10 generics are here. But when you look at the value, it's Abraxane. And Abraxane is 30 times more expensive. Because if you see the unit price of generics is around $30, where Abraxane costs around $900. So doing the formulation right, like in these three examples, I also quoted amphotericin, and I didn't show you amphotericin because generics don't even exist in amphotericin, non-liposomal formulation. Everything is liposomal amphotericin. So formulation is when it does the meaningful improvement, when it fixes the real medical problem, is being really much rewarded on the market. And the key findings and learnings from this case study is very simple. By doing a proper formulation that is fixing the unmet medical need, which in our examples is majority improving the safety profile, and a little bit touching the efficacy. For example, Abraxane has a different indication span than the basic paclitaxel. You will end up with a significant market positioning. You will have market premium, and you will have a market premium, so your price will be extremely above the other options, and you will be in a very nice market position. This is applicable across therapeutic areas. I gave you the anti-infective examples of amphotericin. You also have here the oncology examples, and if we had time, it's a universal finding. So this tells us that even when we do the basic research on the basic compounds very early, be aware that it's not only compound that matters. Formulation can do wonders. Formulation can turn your drug that has a safety problem into a product. It can address some of the safety problems. So it's not just it's important to have product, profile it biologically, and then we will do something, mix the formulation, and who cares, we will go and hit the market. I hope that these examples showed you that there is much more to it, that excipients are very important compounds or partners to the drug substance, and that when you do that right, you can really have a big benefit. And at the end of the day, even more importantly, patients will have a huge benefit. To put it into a more perspective of the early stage now discoveries, there was a very nice survey conducted by InfoPharma, and here I would like to thank Leukocare for providing and sharing this data, where they were asking more than 150 different participants about a very simple question. Have you experienced project failures or a significant delay because of formulation challenges? 60% said yes. Out of these 60%, then they asked, how long was the project delayed? And then you can see that for 38%, the project was delayed due to formulation challenges for less than 12 months. 52% said more than 12 months. And in 10% of the cases, project was killed because of the formulation challenges. Further on, again, an extremely busy slide, which I don't know if you can see, but they were asking what are the key considerations or the value attributes, if you will, when you are doing your formulation trials. And it was IP, time to market, development time, ease of use, of course, IP issues, budgets. So formulation was clearly bringing some of the benefits. And then the magic question was initial versus first commercial formulation. Quite often you do have in examples of many biotech companies that what they do as a first formulation is the so-called initial formulation, which is by far not optimal. And when we know that in formulation you have excipients that are impacting the drug delivery, you can have a bad clinical readout because of formulation, not necessarily because of the drug substance. I'm not saying it's always the case, but it can be the case. So my takeaway and my message to you would be, I don't believe this initial, then you do another formulation, then another formulation is necessarily the right approach. You should do it right the first time, because formulation is the thing that impacts your end game, and it impacts it on so many different angles. So the key findings and learnings from this market survey was that basically, as I just tried to say, that we should do it right the first time. Forget this early stage formulation, then we do the formulation later on, because you are exposed. You are, by doing that strategy, exposed on actual project success level, because as you saw, there are a lot of failures, even project stops due to formulation. But now I will show you where else you are exposed, and it is IP. I'm sure we all know this, but we can in pharma area protect everything, almost the flying insects we are able to protect. So it starts with basically the product, either drug substance or the drug product, also API and finished dose. We do processes, and then it's a whole myriad of method of use patents which are doing dose, specifying the dose, the dosing regime, route of administration, drug combinations, many other stuff. But very, very importantly is that we should all differentiate, and this here we would play a game if we would be in a face-to-face mode. And this is a often and very often misconception that I heard so many times in my professional life, people saying, we are fine, we have our own patent. Like, for example, here you can have a company that has patents, 1 or 2 or 3, and then my examples that I heard so many times would be, we are fine, we have a patent. And I usually then ask those guys, say, really? How sure are you that you are fine because you have a patent? You have a patent, that's proprietary, that's fine. But do you have freedom to operate? And then I starts to roll. But we have a patent, that means we have a freedom to operate. Say, nah, doesn't need to be the case. Yes, it can be the case, like in the example of patent number 3. Clearly here you have your own patent and you have a freedom to operate. In the case number 2, you have your patent, but you have partial freedom to operate. And in case number 1, you have patent, but you have no freedom to operate, because there are some other patents by some other third parties that are either covering parts of your invention or the whole of your invention, or it's broader than your invention. I don't want to go into IP nitty-gritties because it's an interesting story in itself that I'm very passionate about, but they are blocking you. So in this case of 1 and 2, you can kiss goodbye your patent because you are blocked by other people's patent. So yes, you can put your granted patent on the wall, be happy, sing a song, but that's as far as you will go along. You don't have freedom to operate. So don't underestimate that, because there are people coming after you, and these are called generics, and they know that, and they take advantage of that. And just, I would focus on the bottom chart. So if you look, the paragraph 4, that's a typical US game where generic filer is challenging a patent. And then what patents they are challenging, these data are a bit outdated, and I apologize, I didn't have time to refresh them, but basically the green is formulation. The generics are, as a rule of thumb, typically challenging the formulation patents. This is substance patents, this pale color. So the means how generics will enter the market will be through formulation. So even if you don't take the earlier story that formulation adds medical value, patient value, that it will increase your chance of success, it will reduce your time to market and all that good stuff. Take this as an example. Formulation can hurt you commercially and on a patent reasons. I know that for a lot of small biotechs, it looks like, that's another people's problem. I'm very far away from commercialization, and my asset will anyway be commercialized by someone else because it will get through the alliance management through a bigger company. But believe me, bigger company, when it will assess the value of your asset, freedom to operate, IP, will be one of the main value definition in the eyes of your company that you are talking, your alliance manager, because they know they are picking your asset, they need to launch it, and they want to be sure that when they launch it, then they will not face competition left, right, or center, that the generic company will challenge their patent and then destroy the market, that your formulation is weak, that you are blocked by some other third-party patents. And by the way, it doesn't need to be formulation. You can be blocked by freedom to operate or not having freedom to operate on any other patent. So don't underestimate the value of the IP. especially when you are a small company. And then this brings us to, and I think we were here, what are the value additions that formulation can bring? Another important takeaways, and very, very widely used word these days in the public is the value-added medicines, which are in a way drugs that are known. So it's also called repurposing, a little bit different, but fine. It's similar. So you have a known drug, which you then improve the property of that drug and create a new value-added medicine. And you can do that, and a lot of this is sitting on the formulation and on the way how you are improving your delivery. So for example, the first group is where you are improving patient compliance and convenience. Then you can have a much more convenient delivery devices or technologies. For example, if you change a vial with a pre-filled syringe, or you go from a vial into ready-to-use infusion bag, that's very beneficial. You can also have a new formulation, reformulate. You can fix some of the safety concerns. All these previous examples of amphotericin, irinotecan, doxorubicin, paclitaxel, and many others are reformulations. These products have been reformulated post-launch of the brand, and they have been reformulated to fix the real medical problem, which was the safety profile. And there is a big group of improvements you can do on playing with doses, dosing regimes, indications, fix dose combinations, and whatnot. And then this is true and not just fantasy. And this majority of this slide sits on formulation. You can see on statistics, again, I don't know if you can see the years, but it is increasing significantly. The number of filings, it's in US famous 505(b)(2) route, which is used for this, is increasing more than 10% on a compounded annual growth rate. And success rates are for NCEs in phase 3 or those improved formulations. For example, in phase 3 is much more on the side of improved formulations, and also it is to be expected in a way because you sit on a known compound. So with this, I would just like to bring us to the end and link it tight all together. So I strongly believe that we have a big urgent healthcare crisis, which is AMR, and it's causing for disruptions on so many fronts. It's push incentives. I would really emphasize pull. I believe currently pull is probably a bigger challenge than push. And if we now come back to the R&D environment, in R&D environment, we definitely need new compounds. We need new compounds that are active, that are properly biologically profiled, but please don't take CMC and formulation as a foster child, because you can be burned. Like in the examples at the beginning, I'm not saying everything is like that, but even if a chance exists that you will be burned, like in any appropriate risk mitigation efforts, you should avoid it. And also take a formulation at the right time, do it right the first time, because it will add a lot of value. It will add the value that your compound that you maybe suffer with in your profiling biological, you can improve, because you will add excipients to your product that will potentially and maybe help fix some problems, the real biological problems on the efficacy level or on the safety level. And you will be able to get an IP. And more importantly, when you reach the market, either you as a small biotech company or your later on partner, there will be an umbrella of value proposition that will surround your product, that will make it a much better asset. And at the end of the day, if you formulate it properly, which I would like to end, because I believe that's the main part, we are all here for only one reason, and this is the patient and the patients of the world. So if we formulate it right, if we take the CMC and formulation aspects early at an appropriate level, so we answer the what, how, and when questions when the time is right, we will give our patients in need products that will make their life easier, so that their patient experience will not be necessarily the worst, that they will not need, like with some drugs, I know that when you take a pill you need to walk for an hour, for example, and the main demographic population that takes these types of drugs are elderly, which is simply great. You know, you are elderly, barely walking, but because of the how drug is formulated, you need to walk for an hour because of the esophagus. So we really need to take that into account, and because of our patients do the first time the formulation and be in a much nicer place. In the AMR, it's especially relevant because we are in a big innovation hall, and why not use all the armament we have outside of chemistry, fermentation, biology? Let's add formulation and CMC and have a holistic rounding and bring these needed antibiotics to the market. So with that, I would like to thank you for your attention and open up for some questions.
Thank you, Sasha, for this very comprehensive and as usual, entertaining talk. Up to now, we have not received too many questions from the audience, but this is probably because the talk was so fascinating and everybody was focusing on it. However, I want to encourage all participants to ask their questions or share their thoughts via the chat function. And these questions then will be passed through to me, and I will place the questions. So one question came so far from the audience, and I'm expecting more questions to come. This is actually not so much a question, it's more a statement, but it would be interesting for us probably to hear your opinion on this, Sasha. So one participant stated, every AMR pathogen is a biofilm former. Unless new drug candidates address biofilm, they cannot and never will work in humans. Why not do biofilm testing in preclinical trials and save the millions spent to find out that the compound is ineffective? ASTM standard testing methods are widely used. So, Sasha, do you have an opinion?
Yeah, I mean, it's a very good observation and a very valid one. Biofilms truly are a challenge biologically, and also from, let's say, experience that I am exposed to, it is being more and more tested. So people are getting aware of biofilms much more than they were a few years back. So, and they are slowly entering some of the even before preclinical testing panels, which is good news. Biofilms are extremely diverse and very, very biologically intriguing environment. And again, I think bacteria and all these microorganisms are much smarter, quote unquote, smarter, not necessarily brain smarter, but smarter, evolutionary smarter than we believe them to be. And biofilm is one of the ways how they do that. So I do believe biofilm is a big problem. Luckily, also through repair, we have seen some biofilm addressing programs. And also there are more and more attention put to it. It's not an easy problem because it's clearly not a well-defined environment, and it's different from different tissues. But this is something that absolutely should be addressed. And also formulation here, if you do it in a combined way with the effective compound, and if you put something that can disrupt the biofilm maybe in the formulation, and then you let your compound work, could also be one way around it. So absolutely a very valid comment and very relevant challenge for the antibiotic development.
Yeah, maybe to come back to the second part of this question. So what's your opinion about ASTM standard testing methods that are widely used, and why do you think they are not so much employed in or not employed as a standard in preclinical?
I don't believe that's necessarily the case here. I don't believe we have that challenge in the antibacterial R&D. I don't believe is the testing panel. This is probably one of the therapeutic area which has the most well-defined therapeutic, testing panel. So the translation from let's say before in vitro in vivo into humans is not necessarily that bad in the area of antibiotics.
Okay, and you were mentioning that even in the context of biofilms, formulations, and probably specific excipients could be helpful. Isn't that contradictory to what we have learned today and that you have talked about, that excipients are supposed to be ineffective and shouldn't have an effect on themselves? So I would assume that as soon as an excipient supports or provides any kind of functionality towards biofilm formation, for instance, it won't be considered as an excipient anymore?
It's a great question. You were paying attention, but I didn't say it needs to be an excipient. So it can be another compound that you will co-formulate, so that you will have like what we have in the example. It has nothing to do with biofilm, but it's like we have with beta-lactam and beta-lactamase inhibitors, where you have a two drug. In a nutshell, you have a inhibitor of the resistance problem, which is a beta-lactamase, and then you have the antibiotics. So for example, if you combine something that will disrupt the biofilm with even the known antibiotic, because some of the antibiotics really work. The problem is not necessarily that they don't work. They either don't get where they should come. A very good example are the infections of the bone and prosthetic joint infections and bone infections, where we know that antibiotics we have today work, but they simply don't get there. So coming back to biofilm, you can co-formulate, not necessarily with excipients, because you are spot on. Excipient must be inactive pharmacologically. If it has an effect, it's not considered anymore excipient. That's a rule.
Okay, thank you. So there's another question coming in. On a practical front, how does one begin with formulation? Do you begin with the chemical property of the compound?
Yes. The starting point absolutely is understanding the physicochemical properties of the compounds, because as said earlier, you will play with the interaction space, design interaction space between the compound and the excipients. To be able to do that, you need to know how your compound behaves. You need to know its basic physicochemical properties, from solubility, lipophilicity, many other stuff. Is also if you have some structural information. So the more you know, the better. And then you do it in many different ways, but there are more algorithm-based approaches, some are more empirical, some are in between, but it doesn't matter what they are from the approach. They are all trying one way or the other to play with the design space and with the non-covalent interactions between drug and the excipient. And to do that, you need to know your enemy, you need to know your drug. So it starts with chemistry, physicochemical properties of drug.
Thank you. So you claim do it right first time, but this is probably easy said and hardly achieved. When do you think is the right time to develop the right formulation? Because if you're too early, you probably invest in a drug substance without knowing if it works. On the other hand, I mean, as you explained nicely, you also buying in risks to your clinical program if you don't consider CMC topics early enough. So when is, in your opinion, the right time?
I would, in my personal opinion, strongly enter clinics with the right formulation. I wouldn't do phase 1 with a prototype. I would do phase 1 with the right formulation because I'm reading it out. And, you know, doing formulation, this is also a bit of a misconception, is not that expensive. When you look at all the GLP tox profiling that you are doing before entering into clinics, which you need to do to be able to get your R&D approved and enter into the humans, part of that, in my humble opinion, should be formulation, because drug is delivered to a human by the means of drug formulation. And if we are on this slide, there was this linear from substance to formulation to delivery. And if we are going from substance to delivery with skipping formulation, we are doing one very bold assumption, which I wouldn't advocate to do, because you can be fine. I mean, okay, I'm not saying some people basically just took the API like in a vial, put it with some buffer, and off they go, and they ended up with approved product and everything is good, fine. But I do believe that these days are probably more history, because more and more people are smarter and smarter and doing these things. So you are exposed, if not exposed clinically, like in this case, you will be exposed patent-wise, because some other can do a better formulation, file a patent, not you as a brand, and then you will be in a problem.
Yeah, I think there's still this mentality of, before I invest into anything beyond the compound, I want to know that the compound works. But I think you also pointed out nicely that the compound cannot be considered isolated. It's probably also a thing that, which is particularly right for smaller companies who really have maybe not the same resources as big pharma. And the next question goes into this direction. So for a small company without a formulation group, what question do you ask your CRO to make sure that this CRO is appropriate?
I mean, it's not a question. Like anything in life, it's a Chinese menu, and you know, it is also starts from understanding what the CRO can do or not do. So understanding of, ideally you would want to have a CRO that is in the area quite known and focused. All these generalists, except for the fact where you have a huge organization. There are some very big CROs which can do pretty much everything, but typically you want to have people that are specialized in the field, that they have a successful track record. And as in everything with CROs and any external work, what is very, very important is outside of the hard facts, which is abilities, competencies, of course, budgets, timelines, all the hard stuff. But we shouldn't forget the soft stuff. And this is, you know, this partnership that you need to have with your CRO, and which is down to the humans, to collaboration and how the interaction goes. Because this is not something you just sign a contract, give a task to a company, and then you go home and forget about it, and then you come back 6, 9, 12 months after and ask for data. These detached collaborations and partnerships, so that's why I'm calling it partnerships. I don't necessarily consider it as outsourcing. It should really be a partnership, because this is one of your key value driver for the asset, and you want to be in a partnership with that CRO, because you need to share knowledge from your side, and also you need to you can be quite helpful in fixing some of the technical problems that will come along the formulation development, which are, for example, attributed to the drug substance properties. Who better knows the drug substance than you? So this willingness to do it in a partnership way, of course, based on a successful track record, is some of the very, very important decision criteria. So there's not a one. And if you have a small company that is not able to assess that, then take a help, take an advisor, take someone who will help you find an appropriate CRO, who will be able to have a discussion with a CRO on that subject, and then help you select a real one. Don't be just bound by budgets. That's my message. Cheaper is not necessarily, you know, one of the saying is, I'm not rich enough to buy cheap things.
And that brings us straight to the next question. Also, in the world of smaller biotech companies, I think. So the question is, if you would start lead optimization today in a small company, what would be your advice on when and how involve formulation questions, formulation screenings?
In lead optimization, I would just you don't need to do formulation screening at that phase yet, because you are optimizing the compound. But I would have understanding of formulation again some questions asked by someone. Again, if you don't have that someone, there are many people around that can help, because picking the lead, one of the parameters, if you have a choice for, and it's really down to the specific case. Some drugs are having much less problems, some are having much more problems. So I've seen in lead optimization phase, for example, products that were intended for oral use from the medical perspective, and then they had bioavailability, oral bioavailability in single digits. In that case, clearly you need formulation contact in the lead optimization phase, because your compound is a problematic compound, and you maybe need to trade off activity for formulability. Nobody wants an oral drug with a single digit oral bioavailability. It's unformulable. So what I'm saying depends on what specific program you are, what specific chemistry challenges you have, because chemistry challenges will relate in formulation challenges. They do, it's all the same space. It's a chemical space. It's just space between two entities. But having an advisor to just ask questions from time to time cannot hurt.
Thank you. Question pointing towards new modalities. So what are your thoughts on new therapeutic modalities such as lysines, for instance? Do you think...
Such as what?
Lysines?
Okay.
Do you think they would encounter the same challenges as antibiotics in terms of R&D, stewardship, and lack of funding?
That's a good question. I personally think probably yes, because some of the challenges we have is the evolutionary behavior of the things we want to kill, and they will probably find a way how to resist. So I'm not that this is not to the core my subject expertise, but I would say that there could be challenges there. As I said, evolution is something what is causing us problems. And I mean, you see that in pretty much in all modalities I've seen so far, advanced modalities though, you cannot say that resistance doesn't exist. It's just a question when.
Yeah. In your opinion, nanoformulations such as nanoparticles or liposomes, will they be useful in tackling AMR as in the examples you showed for other pathologies?
I mean, they are used in amphotericin case. So so first of all, it's a huge, these particulate formulations is a huge environment, and people call nano many things which are in a way sub-micro. For example, these formulations I showed you, all liposomals were around 200 micrometers and above, nanometers, sorry, and above. And outside of liposomal world, you can have a polymeric particles as well. So amphotericin was a successful example of that. You see now the COVID vaccines are another successful example. Truly, there is a challenge with proper antibiotics, and this is a dose. Antibiotics are highly dosed drugs, like some are even a gram and more. It's not that easy to encapsulate 1 gram of active into any, liposomal or polymeric particle. It comes with the challenges, but I've seen it that it can be done. Some companies manage to incorporate into a particulate system, I think even up to 1 gram. So it is doable, but it's clearly more easier for oncology drug because they are more potent, so less dose, and CNS and some other areas. But I wouldn't underestimate it and wouldn't overrule it, because these systems can fix a lot of delivery issues and a lot of also safety issues, which antibiotics some are having.
Okay, thank you. Another question regarding CMC criteria. So what CMC criteria for every batch should be to ensure consistent in vivo activity, like PK, or what else comes to your mind?
So first of all, documentation should be right. Please make sure that synthetic procedure, especially for the smaller companies, especially for the companies that are spin-outs, please make sure that the procedure you have is reproducible. You can test that very simply. You don't need to wait, like in my examples, when you need more compounds. Just test it. Ask someone else in the team to make compound as per the procedure written by the original person. And just check, did that person receive the same compound with the same quality attribute? This is a very nice, very fast, very efficient way of testing it. So this is the key criteria. Documentation should be right, and you will not be able to assess reproducibility until you try to make it sometimes. And try to keep very well documented starting materials. So always know, I didn't have enough time to show you other examples. People didn't even know where they bought, for example, solvents, which solvent was used, what was the provider of solvent. These are very important stuff because, maybe there will be an impurity in a solvent or whatever. So document, like when you are cooking, don't just put 100 grams of sugar, put 100 grams of sugar from this producer, and even put the batch number. It's just 2 words added into documentation. It's not more than 2 words, but that helps a lot. That allows traceability, that allows reproducibility, and if you have challenges, you know what you've used. So this would be for me #1 criteria, because it starts with that, and then this defines your quality attributes later on.
Okay. A question coming from the audience that I guess you have answered it. I still want to convey it because it's key to your talk. So the question is, talking about early formulation, would you do this prior to phase 2 or prior to phase 3?
I would do it prior to phase 1.
Yeah, I knew you would answer this. Okay. Novel parenteral excipients are very rare, probably due to some conservatism in our industry. Do you think it will change in the future?
Oh yeah, absolutely, and it is changing. Sorry, I lost something as always. We have a specific example as a company where we successfully qualified a novel parenteral excipient. So it is doable. It is not a walk in the park, that I can tell you. It is a lengthy and quite an expensive process, but it is doable. But again, the qualifying novel excipient is almost the same and tougher than doing the preclinical development of a new compound. You are almost doing the same as the GLP, safety and tox profiling you are doing for any NCE, with one bigger hurdle, and then this is excipient, which means there is no toxicity that will be tolerated. For the compounds, because it's an active, agency will tolerate some toxicity signals. As some of our colleagues likes to call it, a novel excipient should be squeaky clean. No signals whatsoever, because this is inactive. So yes, it's doable, but it's like running, have it in your mind, it's like I have another compound I need to push through the GLP preclinical phase.
The next question is interesting because it touches a different route of administration. We have talked a lot about orals and IV. Would you say that inhaled antibiotic formulations are more interesting for lung indications such as cystic fibrosis? And what are, in your opinion, the challenges with inhaled formulations?
Yeah, it's absolutely, I didn't touch on that because of the time, but yes, answer is yes. Cystic fibrosis, not just cystic fibrosis, for example, non-cystic fibrosis, bronchiectasis, for example, and some other indications clearly do require inhaled route of delivery because the infection is happening in the lungs. And delivery challenges for inhalation products are not simple, but they are linked not just with formulation, but they are also very much linked to the device. So what type of inhaler are you using? Typically, the anti-infectives are nebulized, and then there are a lot of nebulizers out there, but there are also some handheld versions of that. So the device, so the delivery device for the inhalation is in the inhalation space probably playing bigger role than excipients, because you cannot have that many excipients for the inhaled products for many reasons, but what will impact delivery will be the inhaler, because you have the usual interplay, how many of the emitted dose enters into your lung. So what's the administered dose versus emitted dose? And just as you have some products on the market that have only 10%, which means you are inhaling X, 0.1x is in your lungs. The good inhalers are increasing that 10% to some 30, 40, 50% of the administered dose ends in your lungs, but it's pretty much governed by the inhaler. And there are many inhalers, and this is where drug and device work together to have the desired delivery.
And what's your expertise with reproducibility of bioavailability, for instance, if you go the inhaling route of administration? Because I think, obviously, if I go IV, bioavailability is pretty much straightforward. With inhalation, obviously, that's another piece of cake when it comes to reproducibility.
Bioavailability by inhaled route is a tough cookie, and that's why you can see that very few, but this is more applicable for the follow-on products that are then using this route to basically compare to the existing brand, and very few manage to get a product approved by the bioavailability requirements. If you don't meet the bioavailability requirements, you always have the clinical endpoint, phase 3 type of clinical trials, where you can, in the case that bioavailability fails, if in your clinical endpoint study you meet the requirements, you will get your product registered. But you are absolutely right, or whoever asked that question, it is reaching bioavailability with inhalation is tough. But don't be discouraged if you fail. You can still get your product registered. It will just cost you. You add 1 0, if not more, to your clinical program, and you add some years and uncertainty.
This is somehow encouraging, I guess. Next question. Let's assume that one discovers that 2 old drugs are unexpectedly synergistic. So one could think to formulate these 2 drugs in one combination product. Can you comment how you see this product from an IPO, FTO point of view?
You need to look at a specific, you absolutely need to do the FTO search, number 1. So you need to look what's the IP environment for drug 1, for drug 2, on all the pyramid patents, not just on the level of product. You need to search for everything: product, process, use, all 3 branches, and you need to find what's out there, because co-administering in a one tablet, if there are already IP around it, could be a great product, but you will not have an IP protection for it. So it really needs to be, it starts with a thorough and detailed IP freedom to operate search, and then based on what you get from that search, then you can see, do you have a space or not? So it cannot be a theoretical answer. It's a very practical case-by-case answer.
Okay. So maybe last question for today's webinar, and this is directly addressing your role within Xellia. Xellia has not listed any new entities. All testing is done on new formulations. Is this a dogma for Xellia, or are you going to change it?
No, we are, first of all, as a scientist, we don't follow any dogma. We are critical thinkers, so there is no dogma. We strongly believe that some of the products we work on do require reformulation, and that's why we do it, and we do it with patient in mind. And we do believe that by doing that, and we have some great examples of successful stories where we have done so. So we start with the patient, we start with the patient need, and then we look at the product that is given to a patient, granted, currently a known one, how can we improve it? So absolutely not dogma, it's how we make products more in a more compliant, convenient, and patient-friendly way given to a patient that then at the end of the day have a much better therapeutic effect. And this is why we are all around. I mean, not for everything new drugs are needed. And that's the beauty of antimicrobial search. Like, you know, with clothing, you don't need new shoes every year. Maybe a bad example, but doesn't matter. You need really to fix the real problem. So no dogma, it's a science, patient-driven approach.
Okay, thank you, Sasha. So I think I've canceled my super sophisticated outro in order to give you as many questions as possible. Unfortunately, we couldn't answer all questions, but just the pure amount of questions could show that there's a huge interest in the audience, and I can just encourage everyone who took part today to reach out to Aleksandar to get more insights into this fascinating topic. So from my side, thank you, Sasha. Thank you very much for this inspiring talk and the interesting answers to multiple questions. And thank you to all participants. I think it was very exciting. And having said this, I hand over back to Victor and wish everyone a nice rest of the day wherever you are. Thanks.
Thank you also from my side to all the audience, and very good questions. I'm sorry I probably didn't answer all, and thank you, Michael, for moderating.
Yes, thank you, Michael, for moderating today's Q&A. And of course, thank you to Aleksandar for giving us valuable insights into CMC and formulation aspects in antimicrobial R&D. So the registration for our next webinars will be soon announced and open for registration on the website revive.gardp.org/webinars. So please make sure to keep an eye out for our emails and to stay up to date on our future webinars. So that will be all on our side. Thank you very much for joining today, everyone, and for contributing to this discussion. I really hope you found this webinar interesting and useful, and that you will join us again for our future webinars. Please make sure to spread the word in your network and to encourage your colleagues to join as well. Thank you and goodbye.